And we're doing a very careful study of different enteric infections these children suffer from to try to see is there like one that's more likely to be associated with stunting. Because maybe we can make a vaccine to prevent at least some of malnutrition. You always want to know what your kids think what you're doing at work. And Danny, who's now in law school, when he was in 3rd grade, was asked to draw a scientist at work. He wrote, well, my scientist made a mistake. And you see the uh-oh, the explosion, and the blackened scientist. So I guess that's what he thought I did at work. My youngest son thinks that all I ever do at work is have parties and eat, because the only time he's ever there is like when we're having like, a lab luncheon or something. So what your children think you're up to. But you sort of hope that when you're doing this work that you're not making a mistake. And so we've set up this whole cohort to study malnutrition, and in fact are the children becoming malnourished that we're going to have the opportunity, because it could be by giving them free medical care, by visiting them in the homes and teaching them about oral rehydration for diarrhea, that they won't become malnourished, and that this whole study would be a big mistake. And you can say, like, fortunately or unfortunately, that was not the case. And so when we looked at children when they were born, they were almost a standard deviation below the WHO norm for height for age. And you notice by 15 months of age, the average child is now moderately stunted. For weight is a little bit different. There's a little bit of catch-up, and then, again, like this inexorable decline. Again, like this, it's so impressive to me what a difficult problem this is, because these children have better access to medical care than probably children in Athens. One of the things we instituted under the recommendation of Cynthia Snyder, who's an MD and has an MPH, was, and this should probably seem obvious to everyone in the room except for me, that it wasn't ethical to follow children into malnutrition and do nothing about it. And so when children do become malnourished, we actually have malnutrition clinics that we've established with the support of the Gates Foundation in Dhaka, and the mothers bring their children in for supplemental feedings. And this is actually fairly popular and adhered to by the moms, because it's also an opportunity for them to get together and chat as their children are getting a nutritional supplementation that includes like this lipid-based nutritional supplementation I told you about earlier. So again, to just complete this thought about the gut being abnormal, normally your gut is very good at keeping bacteria from getting into the systemic side of your body. If the gut becomes leaky due to chronic inflammation, then you begin to develop antibodies against a bacterial toxin called endotoxin. And so we measured that in the children, and what we discovered, again, was the children who were most severely stunted had much higher levels of endotoxin antibodies, again sort of suggesting that maybe this sort of core problem that's going wrong is that their gut is abnormal. So again, there's been like many students at this site working with me. This is Tanya Thomas, who's a pediatrician and an internist and an infectious disease fellow. Saria Izeldin, a medical student at UVA. Grace Malad, a medical student. And Katie Ralston, who's a postdoctoral fellow, all from UVA from 2 summers ago. One of the adjustments of working in Bangladesh is that Friday is Sunday, and so everybody works on a Saturday. So, but I understand like that you guys are all working on Saturdays too now after like the big snowstorm and things. I heard that that's like. So I wanted to say, so now I'm going to get into the genetics, and a little bit of a show of hands here, because we're supposed to have, like, I think one of the big things with education now is active engagement of the students, and so that you're not passively listening. So I'm going to ask you to first vote on how many genes make up a human being. And I'll give you one hint, is that E. coli, the bacterium, has 5,000. And so everybody's got to vote. And so raise your hands. How many people vote for 20,000 genes in humans? Okay, it looks like about 20 votes there. So how about 40,000 genes? So 8 times. So a little more votes for 40 than 20. And how about 100,000 genes? Okay, so smattering of votes for 100,000. So that's actually very good. The correct number is 20,000. But if you voted for 40,000, that's what people used to think just like about 3 years ago. So you're not like very, very far off. And that's in its own right is pretty amazing, isn't it? That only 4 times more genes than E. coli are one of the things that make us what we are. So one other thing I'm going to ask you to vote on is how different is each one of us in this room? So if we sequenced each person's genome in this room, which we can do today with a royal we, I couldn't do this, but for $5,000 we could sequence any person's genome in this room. And if we did that for 2 people, would there be a difference in sequence every 1,000 bases, every 10,000 bases, or every 100,000 bases? So just how different are we? So how many people vote for 1 in 1,000 would be different? All right, so about 10 votes for 1 in 1,000. How about 1 in 10,000? Okay, about 5 votes. Everybody's got to vote now. So how about 1 in 100,000? So most people vote for 1 in 100,000. This to me is like, this is like a totally amazing thing. It's just how different each of us has been created. And now you can say, well, a lot of those changes probably don't make any difference. They're probably in so-called junk DNA, and that's true. But a lot of these changes are going to be in very important genes, and we really do differ tremendously, in our genetic makeup. Like I always remember having a friend. He always told me, "Well, I never get the flu," and I didn't believe him. And he may not have been telling the truth, but it could be that he never does get the flu because he has a genetic makeup that makes him intrinsically resistant to influenza. And so the real question we're getting at is could some of these 1 out of 1,000 base pairs make some children resistant to becoming malnourished under a setting that most children would become malnourished? So there's 3 billion base pairs. So again, if you sequence any 2 individuals, we would differ by 3 million individual bases. And there's also even more copy number variants where parts of your genome are amplified or are deleted. So huge amounts of variation. Of course, now there's 3 million differences between any 2 individuals. It's not the same 3 million if we sequence 3 people. And so when you actually look at everyone in this room, there's probably about 25 million sequence polymorphisms or SNPs that are represented in this room. And now some of those are going to be common. So some are going to be present in 10 to 50% of us. Others are going to be less common, like maybe 1 person in this room will have it. And some will be private. And so it'll only be a SNP that maybe is in your family, maybe just shared with your sister or your brother. So tremendous variation. And what's happened in the last 10 years with the sequencing of the human genome is the ability to use this variation as a way to ask questions such as genetic differences that could control susceptibility to malnutrition. Now we had a big advantage in that the Bangladesh children are very closely related to Caucasians, which are in green. And so this is what's called a principal component analysis, and we've looked at 1 million different differences in the genome of the children in Bangladesh, which are in red. And we've compared them to Gujarati Indians that were tested in Houston. That's where the GIH comes from, as well as Japanese, Chinese from Beijing, the Yoruban tribe from Nigeria. Well, the fact is that since these children are close to Caucasians, was very helpful to us 3 years ago because all the genetic tools, of course, had not been designed for Asians or Africans, but for Caucasians, because that's where all the interest is in medicine, with coronary artery disease and so on. But the fact that these children were closely genetically related to Caucasians allowed us to use tools 3 years ago, which would not have worked if it had been an African population. And this is the study that I'm going to explain to you. It's called a genome-wide association study. So you look at here, this is an area on chromosome 9 that's shown here. And here's an example of a single nucleotide polymorphism. And so one individual has a G, C, and another individual has a TA. And here's a second polymorphism, AT to GC. And in this example, the gene of interest is going to be between these two SNPs. And it's a very simple case control study, so you really don't have to understand anything about genetics to do a genome-wide association study. Because it's a very simple question. If you look at your cases, and so in our situation, this would be children that are malnourished versus children that are not. If you look at like SNP number 1, and if you notice like this SNP number 1, which is this T to A, is more common in the cases, and I think that's the orange than in the controls. You notice like there's more orange people in this group of like 1,000 individuals. Same thing with this SNP as well, more common in cases than in controls. And so what this is kind of like a mile marker on the genome, is that you know like, well, there's something different about the DNA in this region in cases of malnutrition than in controls, as an example. And the way that you measure this is just with a p-value. And so, and again, this is an example. This is not our data for malnutrition. We're looking at the p-values. So from 10 to the minus 2 to 10 to the minus 14th, and this is each of the autosomal chromosomes, 1, 2, 3, 4, and so on. And what you'll notice is that for the most part, there is no significant association of p-values with the phenotype that they're looking at. This is all just sort of background. But then here's this peak right here that represents like these two single nucleotide polymorphisms where there's p-values of 10 to the 10th, minus 10th, 10 to the minus 12th. And so this is an area of the genome that is significantly different between cases and controls. And this is sort of the basis of a genome-wide association study. So it's very, very simple. You just measure 1 million single nucleotide polymorphisms, and for each one of those million differences you ask is one more common in children that are malnourished than well-nourished. And so here's our data for one of the genes. And so we're looking at a position on chromosome number 2. Here's the actual base pair numbers along chromosome number 2. These are p-values of association of individual SNPs. Each one of these little dots here is an individual SNP with being malnourished. And so you notice for the most part this is all noise, but then here's a peak right here where the p-value is 2 times 10 to the minus 7th. And you notice that there's a series of SNPs also that are all significantly associated with being malnourished. Well, the beauty of this analysis is that these SNPs are so densely located in the genome. This isn't showing you a part of the human genome where there's 500 genes. This is showing you an intron in one gene. And so you go instantly from measuring 1 million SNPs knowing what genes are different between children that are malnourished and well-nourished. And this is the preliminary results. This is just from November, and it's work that's still in progress. And what I'm showing you is the name of the gene. A couple cases, you notice that we found an area of the genome where there is no gene but it's still associated with malnutrition. So there must be some non-coding function of the genome there that's important. Here's the chromosome number. This is the effect of having this SNP on your height for age. And so if you have a protective SNP in this gene, your HAZ score is 0.4 standard deviations better. Remember, it only takes 2 standard deviations to take you from well-nourished to malnourished. So this single change is having a major impact on your nutritional status, or is associated with a major impact. And you notice it's very common. So 12% of the children have this change. So we're not looking at rare changes in the human genome. We're looking for common things that could explain this very common problem of malnutrition. And then the other thing I want to point out that was so interesting is that all of these genes are involved in lipid metabolism. This is an LDL receptor family member. This is phospholipase D1, which is involved in catabolism of phospholipase C. And this is the HDL binding protein. So there are changes in 3 genes that are all involved in lipid metabolism that are associated with malnutrition. You remember like that one of the bright lights on the horizon is lipid supplementation. And it leads us to wonder perhaps if maybe one of the differences that protects some children from being malnourished is changes in their lipid metabolism genes that allow them to survive on fewer supplemental lipids than the average child. And so just a little bit of data about one of the genes is the LDL receptor, the phospholipase D1, and the HDL binding protein. So where we are right now then, we're very close to genome-wide significance for these. We need to accumulate additional children before we would publish these results. So I'm really kind of showing you the work that's in progress.